Multi-omics might be very useful in understanding this mechanism of progression from a non-tumoral premalignant status to overt Waldenström macroglobulinemia, because it studies the problem, the issue from different point of view. For example, in the grant, in the program that we are running, the Robert Kyle Award, we are looking at this issue by three tasks. First of all, by studying mutations, different gene mutations in premalignant, and then comparing them to Waldenström sample from Waldenström patients. So mutation, a classical panel of mutations and additional mutations that are not classically studied. Then we study the RNA, so the expression of genes involved in the progression of these malignancies. And we will study RNA expression in cell-free RNA, so in plasma, in a simple blood drawn. And finally, we will study the mutations of the non-malignant counterpart of the cells present both in IgM gas and Waldenström by studying what is called CHIP, that means clonal hematopoiesis of indeterminate potentials. So the mutations in the non-malignant cells that might contribute to inflammation and to progression of a non-malignant condition to a malignant condition.